This is me.
Okay, so we're starting part A of the AP Photosynthesis Lab. And you can see all the materials that are lined up. We've got some colored cups, we have syringes, we've got a bicarbonate solution, and then we've got kids that are already starting to cut out leaf chads out of the spinach leaves. So they need 15 chads per setup, so it's going to take a little while to do this. Okay, so they have part A up and running. You can see the lamps. So what they did is they used little baby beakers, and underneath each of the colored cups in the clear cup, there is a, you can see it right there in the clear one, there's a beaker filled, isn't that cool, with the spinach chads. And so the light is on, and they're getting some good data. Do you guys have any floaters yet?
Yes.
Okay, they've got some floaters. Look at this. The blue cup and the clear cup has some nice floaters.
The red has some. Green has none. Yeah.
Okay, green has none. Okay. And while they're doing that, they're over here. Okay, so we are finished with part A. Look how beautiful. We have the light. Discs have risen. So here is your data. We measured every 2 minutes, as you can see in the chart. So you guys will need to copy this data down. And then once you copy it down, you need to calculate right here the ET50 and the 1 over ET50. Remember, ET50 is the estimated time at which 50% of the discs have risen to the top. So we use 12 chads, so we want to know the estimated time at which 6 have risen. So for this first one, you can see between, right here we've got 5 and then we have 7, so 6 would have risen between 10 and 12 minutes, so the ET50 would be 11 minutes for the red. If we go over here, you have to set up a proportion to figure out this one, because at the end of 36 minutes, only 5 discs had risen. So you have to say 5 have risen after 36 minutes is to 6 discs after X minutes, and you set up a proportion up. Okay, this one is pretty easy. 6 discs have risen after 14, and then this one is somewhere between these two, which is 13 minutes. So you have to do this one, calculate that, and then the bottom is do the inverse of that. Take your calculator, 1 divided by ET50. And that concludes part A.
That's a pretty good line.
Okay guys, so this is part B of the AP Photosynthesis Lab. This is the chromatography. So we've got these vials that they're going to add some chromatography solvent. If you look right here, chromatography solvent. They're going to add a little bit to the bottom in a second, but right now, look at this magic. They have 2 pigment lines. One of them, you can see kind of like the reddish color. Look at that. That's a fall leaf. And then this beautiful, look at this spinach leaf. So down at the tip, they used a coin and they rubbed the leaves to create that pigment line. All right, go ahead, yeah.
Where is the pipette?
So the pipette, let's see where the pipette is.
Fill it up like to this line.
Okay, so we're gonna add, you can see, perfect using pipette with chromatography solvent. Add a little bit to the tip. Okay, perfect. Because we want the pigment line to be above the chromatography solvent. So here you go, here's the first one. That's the fall leaf, beautiful. And so now they're gonna, ooh, look how good that is, y'all. You can actually already see the solvent line moving. You can see the solvent line is the clear, kind of like liquid moving up the paper. So they're gonna let this run for a while. It'll probably take about 15 minutes. We've got the second one filling up now. And then this is where the spinach leaf is gonna go. They have beautiful AP students doing this technique for y'all. I think that's enough. Yep, that's probably perfect. Look at that solvent line, how fast it travels in that one. Okay, so there's a spinach leaf, and they're going to cap them, let these run. Be careful not to tip them over.
Already gone up.
Okay, see if you can see that solvent line. See, it's easier to see it with that light behind it. Isn't that cool? Yeah, I can see it moving. Can y'all see it?
Negatory.
Solvent line. And that is a perfect pigment line. I think you're going to get all the pigments come out. Good.
Yeah.
Okay, so we'll let these run for a while. We'll come back. Okay, so here's the end. We've got both of them. Ooh, that one's a beauty. So they're going to mark the solvent line on the one with the pigment, the spinach pigment. The other one you're just going to use to look at. So go ahead and put that one down. Take a pencil and mark where your solvent line is. Let me get the
I guess it's like right there.
Yep, perfect. Make a little mark. Very good. Okay, so now the rush is over with this, right? So in the lab, if you see right here, you're going to be using the formula to calculate the RF value. So the first thing you have to do, use your ruler, is measure the distance in millimeters from the, I would do the origin, from the origin to the solvent line. So take your ruler and go from your origin of your first, the base pigment, your pigment line. Just flip your ruler around so we use millimeters. There you go. There you go. Perfect. So we're going to go up to that solvent line. What do you calculate?
So like 8.5.
About 5? Okay, so that's centimeters, so we're going to go 85. Is that about right? What do y'all think, virtual kids? I say 81, 2, 3. I say 83. 83. Okay, millimeters, so we can record that somewhere. Yeah, just write that down. So that's the distance from the origin to the solvent line. Okay, now the fun part, you guys get to take your pencil here. I'll show you the first one, then you can do the other ones. You can see this different color green right here at the bottom compared to that more of a bright green. That is 2 different pigments. So you want to mark the base, like let's say it's right here, of the first one, and then we want to mark the base of the second one, which is kind of like right there. Okay, you see that? And then you can see another pigment. Do y'all see this one right here?
Yes, ma'am.
So that one will be about here, and then, the one up here, that's your last pigment. So you mark that. Okay, so now according to the formula, you want to count from your origin to the base of each of those pigments and then record that in your data table. All right, so we're going to calculate the distance from the origin to each pigment. So my origin is going to be right here, the base of the pigment, and we're going to count up to the first pigment line. All right, so you guys ready? This is your first measurement. Okay, so we're going to go, let's see, I guess I should go like this. Silly goose. All right, there's the base of your pigment. You're going to the first pigment line. To me, it looks like 5 millimeters. Okay, the second one, let's say, let me see, 7.5 for the second one. The third one, 15, 16, 17, 18, 19. And then the last one, that's 1, 2, 3. Okay, let's see if we can get it. Okay, the last one up there is 65, 66, 67. Looks like 67.5 for that last one. Okay, so this is part C of the AP Photosynthesis Lab. This is stomata exam. So we're going to be examining the stomata of 3 different species of plants here from Kingwood, Texas. The first one is rose. So I have these stored in water to keep them fresh. This one is lantana. You can see what they look like. Leaves look a little different. And then this one is dianthus, which has little short leaves and some pretty flowers. Okay, so dianthus, lantana, take a look at that, and then rose. Okay, so the next step is we're going to use clear fingernail polish and paint the back side of the leaves. Perfect. So that we can get an impression of the stomata. We go along, we paint them all. Perfect. And then we're going to let these dry so that we can use tape and pull off an impression of these. So she's doing a great job of moving along and painting the back so we can let these dry. Amazing. Good job. Okay, so now what we're going to do, our nail polish is dry. We're going to take a piece of clear Scotch tape right here. We're going to stick it where our nail polish is on the leaf, like this. make an impression. And now that we have the leaf stuck on, we're gonna peel the leaf off so that we have an impression of it. Okay, so this is my tape. I pulled it off the first leaf, so it leaves an impression of fingernail polish, like this. We can make a dry mount by just sticking this onto our slide, like this. We stick it on, look at that, perfect little dry mount. And then we take this slide and we put it underneath a microscope so that we can try to find the stomata. So we're gonna take our first slide, this is Rose, and we're going to examine it under the microscope. The first thing we wanna do is make sure this is on low power. It's the yellow objective and it's 10x. You can see on this side, I can zoom in right here. It's got a 10 right there, it's 10x. So we're gonna put this on the stage. Use our stage clip, there we go. I'm gonna put the stage all the way down and look through the ocular lens and raise the stage up until it comes into view. Okay, so this is gonna be your first field of view for the rose. You're gonna wanna push pause to this video because I'm doing my best to get it centered. Where you see the pointer, that black line to the left is pointing at one stomata. So if you push pause, you should be able to count all the stomata. Let me see if I can get it one more time. This is extremely difficult to do. Hang on one sec. Okay, there you go. So push pause. Hang on. There you go. Push pause and count.
Oh my gosh, I just customized our group chat. Now it looks like
yeah, well, I'll make a group chat.
There you go. Okay, so push pause, count, and make your drawings. That's Rose. Okay, so I just finished the rose. I counted 3 fields of view. So the first time I counted, I did the 7, and then I moved it over, kind of right, left, up, or down. I counted 8, and then I counted 5. So that was the rose. And then you have the picture through the microscope to do your drawing. So you need to come down here, and in your lab book, you do a drawing of what you see. Follow all directions on how to label and coloring or shading using colored pencils. And don't forget your total magnification at the bottom. And remember, I used the 400, which means it's 400 times, or 40x, but it's 400 times because the eyepiece is also 10 times. Okay guys, here's the next one. This is Lantana. You'll need to push pause on the video so that you can make your drawing. I have the pointer on one stomata. I'm sorry this is moving around. If you're motion sick, you need to push pause. Hang on, one sec. Oh, come on, there you go. All right, there you go. That should allow you to make a drawing of the stomata. Just like I said, push pause so that you can get it steady in motion. Ugh. There we go. Okay, but as far as the number, I'm gonna give you the count. Okay, here are your counts for Lantana. Field of view 1, 2, and 3. And now all we have left to do is the dianthus. All right, here's the last one, guys. This is dianthus. This one's a good one. Remember, push pause so that you can get a snapshot. There you go. Okay, and your numbers for dianthus are right here. Field of view 1, 2, and 3. So what you need to do now, okay, you're going to take all 3 of these. You need to calculate the average number in all 3 fields, calculate the average, and then use the lab data sheet, the directions, and calculate the average number per millimeter squared. And then in your lab book, you're going to have a picture for each of the 3 leaves. Don't forget your title and your total magnification below that.